So this is the second part of the Chapter 15 lecture. So this lecture is going to look at transport through vesicles. So movement from the ER out to the surface of the plasma membrane. Part 1, if you haven't listened to, looks at gated transport and transmembrane transport. So gated transport being cytosol to nucleus, and transmembrane transport cytosol to the ER lumen. So this continues the journey of the protein from the ER lumen to the Golgi out to the surface. So as I just mentioned, we have these 3 different ways, and so this audio lecture focuses on our third, where we're using vesicles to transport. So with vesicle formation, we can have 2 different transport mechanisms. So we can have exocytosis. So with exocytosis, the vesicle is going to contain material that needs to be secreted to the outside of the cell or transported to the plasma membrane. And so the membranes of the vesicle will end up fusing with the plasma membrane, and the material again will either be secreted or it could be embedded into that membrane and retained in the plasma membrane. With endocytosis, this is the process basically in reverse. And so with endocytosis, this is where a vesicle is formed. And so material from the outside or material that's attached to the plasma membrane is brought into that vesicle, and then that vesicle moves into the cell. So vesicles are used to shuttle cargo from one intercellular component to another. So vesicles are always not destined to go to the plasma membrane. This is where we can have movement from the ER to the Golgi, or from the Golgi to an endosome, or such. Okay, and so we are going to have this component that's going to donate part of its membrane and proteins and material that are contained in its lumen. And what happens is that you get this budding off, and then we're going to have a target component. And so with the target component, we then have fusion of the membranes from the vesicle with the membranes of the target component. And so proteins found on both the vesicle and the target component allow for this fusion to happen, and we'll look at that process where snares are involved. So this figure shows a summary of vesicle trafficking pathways, and so we just want to keep in mind that we're not just talking about movement of material from the endoplasmic reticulum to the Golgi to the cell surface, that we have other vesicles that are involved in this process, and we also have material that's moving from the cell surface back to the Golgi, okay, and then from the Golgi back to the ER, okay, so we can get this recycling of membranes occurring. So vesicles know where to go because labeling that occurs on them. So we might look at a vesicle and say, oh, they all look the same, but they actually are going to have proteins on their surface, and those proteins that they have end up telling the vesicle where it needs to go. So we can have Clathrin-coated vesicles, COP1 and COP2. So COP stands for coat protein, and so there's coat protein 1 and coat protein 2. And so depending upon what is on the surface of the vesicle, it would then determine where it goes. And so our next figure here, this figure shows you the different trafficking patterns that can occur based off of the different vesicle type, okay, in their labeling. So we can see with COP2, we can have movement from the ER to the cyst side of the Golgi apparatus, okay. if we have COP1 movement would indicate that we would go from the trans side of the Golgi to the cis side of the Golgi. Okay, where Clathrin-coated vesicles, this is where we would have initiation of endocytosis. Okay.
You carry out.
So I'll post a video, that video up onto the Unit 2 files. So with as you saw in that video with Clathrin-mediated endocytosis, we're going to get this budding in of the, it will initiate the budding in of the plasma membrane to create a vesicle, and then we're actually going to get this kind of pinching in that allows for the vesicle to be freed from the plasma membrane so that then it can move into the Golgi. So this is looking at Clathrin-coated vesicles, and so they kind of have this pattern appearance to them, where they almost look like a miniature soccer ball. So I'm going to go to the next slide. So if we look at this under high resolution, we can actually see that we have this like heavy and light chain that are interacting together to create a subunit. So we're going to end up having 3 of these heavy chains interacting with 3 of these light chains to give us this Clathrin form that has this resemblance of 3 legs. Okay. Okay, so if we look at this assembly that's going to happen, so we're first going to get coat assembly and cargo selection. So this is where the adapter protein is going to assemble with the plasma membrane. Those cargo molecules are going to be binding the receptor. So this starts to initiate the budding formation. The Clathrin is going to bind to those adapter proteins to cause that further budding and the vesicle to be formed. We're going to end up getting kind of this neck that is going to be produced. So dianin is going to actually cause there to be the squeezing in that neck, that kind of neck area that pinches in, so that the vesicle will pinch off and be free. Once the vesicle has directed to the location it needs to go, a non-coating process can happen where the Clathrin molecules dissociate and they can be recycled again. So we're going to look a little bit more at this, the vesicle pinching off. So here in this figure we can see where that dianin and associated proteins are going to be wrapping around. So we can see in that electron micrograph how there's that kind of coil going around the neck of the vesicle. Again, allowing for the vesicle to be free so we can have endocytosis occurring. Okay, and so if we have mutations to dianin, this is actually going to cause deficiencies in vesicle formation. So this process of coat recruitment is regulated or assisted by GTPases. So GTPases are going to provide the GTP, the energy which will allow for these subunits of Clathrin and coated proteins to come together to form that kind of cage from it. So remember we saw GTPases when we looked at nuclear pores and the ability to move substrate material in through the nuclear pores. You know, we need some kind of energy in order to do this, and so it's not just ATP that does work with inside a cell, but GTP allows for that to happen. And so just keep in mind GTPases have a role in the transport in both the first pathway movement into the nucleus, but also in this third pathway where we're going to have vesicle formation. Another way that vesicles are targeted to their location is through which Rab protein is present. So Rab proteins are another GTPase. So they work together with SNAREs, and so depending upon the Rab protein that's present on the vesicle, it can control where the vesicle is actually going to end up going. So you can see in the table the number of different Rab proteins and what location that they would be found in, and you can see that there's different patterns depending upon what organelle you're looking at. Okay, and functionally they're going to be similar to the SAR GTPases in their function. Okay. So I don't expect you to memorize all of these different Rab proteins. You should just be aware that Rab proteins are another way that we can have vesicle targeting, just like we can have Clathrin versus coat protein 1, coat protein 2. So all these different factors allow for vesicle targeting. So the Rab proteins are going to work together with the Rab effector, which is a tethering protein. So when we have our targeting membrane, as we can see in this figure, it's the Rab GTP that is going to bind to the Rab effector, and that's going to tether the vesicle. And then what's going to happen is docking. Docking is where you are going to have your V snare, which is your vesicle snare, and your T snare, which is your target snare, are going to become intertwined, and that's going to like really anchor that vesicle down so that we can then have fusion of the membrane. Okay, and so again, the snare proteins are really important in this process, just like the Rab proteins are too. Okay. So we can kind of think of this as, the vesicle is like a boat, and when it's coming into dock, a lot of times someone will be out there on the dock and they'll throw a rope to someone on the boat, and the person on the boat will grab it, right? And so the person throwing the rope, that rope is that tethering protein, the Rab effector. The person on the boat that's going to catch the rope is the Rab GTP, and so they're going to kind of pull that boat into the dock. And then, on the dock, you're going to have, and I don't know what they're exactly called, but they look like little T's that allow for you then to tie the rope from the boat onto it, right? And you knot it, and that really ties and it secures the boat to the dock, right? So that's what the snares are like. The snares, the rope coming off the boat. The T snare is that T-like structure that's on the dock that the rope gets anchored onto, knotted onto. Okay, and then we have fusion of the membrane. So whatever was on the boat, the cargo, is offloaded onto the dock. Okay, and it's a fusion of the membranes from the vesicle with the target membrane that it has now docked with. Okay, so here we can see how we have our snares coming together, so they end up again intertwining and fusing together, so that those interactions can occur. So what's going to end up happening is when we look at the membranes are going to form a stalk. So remember that these are bi-layer. So the phospholipid layer of the vesicle is going to fuse and form a stalk with one of the top layer, the extracellular layer of the plasma the plasma membrane, but the membrane of the target membrane. That stalk gets elongated, so we end up with this hemifusion between the internal parts of the bilayer, and eventually that hemifusion fuses together, and you have basically a breaking or fusion of the two membranes together, so that now there's a connection between the vesicle and the target membrane. There's further kind of, if we think of budding coming, this membrane's coming off, now we're getting like a debudding where they're going to fuse together. Okay, and so this process of vesicle fusion utilizing snares has been well characterized. So back when I was an undergrad, it was like a new hot thing about snares and like, what are these proteins? They've been looked at largely in neurons, and that's because of the release of neurotransmitters. So if you remember back to our chapter 12 lecture when we were talking about the release of neurotransmitters, this is the release of neurotransmitters is all through vesicle formation, but then deformation, right? And so if we block the release of neurotransmitters, what we're going to be doing is blocking the snares, right? Because if we're undergoing exocytosis, it's just the reverse of endocytosis, and we'll talk about that a little bit later. So if you were to give a neurotoxin like botulism, they actually cause cleavage of the snares. So that docking might happen, but the actual anchoring does not happen, so you can't get fusion of the vesicles with the membrane. And so because there's no neurotransmitter being released, that's where you get paralysis of death, right? If you had exposure to botulism at high amounts. So obviously if you're having Botox done, that's a low concentration of botulism toxin. So you're paralyzing the muscles that are being injected, but it's not going systemically to cause issues. Okay. So in order for this, for the snares to dissociate, so they unravel and are reactivated, there's a protein called NSF. It's a sensory fusion protein, and so it allows for the dissociation of your vesicle, your V snare, and your T snare, your target snare. And so your target snare, your vesicle snares can be reincorporated into another vesicle. So the process of membrane fusion is also looked at in virology. So there's a similar system that viruses use to gain entry into host cells. So again, they're kind of similar to snares. And so HIV is a virus that infects CD4 positive T cells. So it uses the CD4 receptor that's on the surface of the CD4 positive T cells. And the CD4 binds to GP120 that's on the HIV. So GP120 is glycoprotein 120. And so that causes that kind of tethering effect. So that draws the virus in closer, and then it has these fusion proteins that are very much like snares. It undergoes conformational changes when it binds and inserts into the membrane, and then they end up fusing, twisting, and this, like the snares, ends up signaling to allow that vesicle to enter in, and then that, because of the fusion of the membranes, you end up getting entry of the viral genetic genome. So nucleocapsid is the viral term for the capsule that contains the nucleic acid of the virus. Okay, so you can see in the electron micrograph images below this process occurring. So next we're going to look at the process of moving material through vesicles through the ER to the Golgi apparatus. So we want to keep in mind that it's only correctly folded proteins that are going to leave the ER, right? So there's going to be these molecular chaperones that are going to assist in making sure that the proteins are folded correctly. Resident ER proteins, proteins that have the KDL sequence that says retain, keep me here, will remain in the ER, but it's the ones that do not have this retention sequence that are going to then be picked up by receptors and moved into vesicles so that they can bud off and move into the Golgi so that we can kind of move to the next step, right? So further processes is going to be done. So signals that will say, don't keep me in the ER, and many times end up getting blocked. So scaffolding proteins like BIP, this is a immunoglobulin binding protein, also known as GRP78, which I've mentioned. So they're going to, BIP, GRP78 are going to assist in the folding, but when they're holding the protein and helping it fold, it's actually going to mask that leaving signal. So during that time, while the protein is coming together and being folded, it actually looks like it should be retained in the ER because, again, it doesn't have this exit piece. Once the protein gets folded correctly, so here we can see in this figure we have this complete antibody. We have both heavy chains and both light chains. So now this is a functional antibody. So now it can be secreted. And so you can see how BIP has dissociated. It's no longer bound to the antibody. So now the exit signal is available to be recognized. And so it's going to be moved into a vesicle so that it can be transported. So just like the snares are important in vesicles moving around, they're also important for the vesicles that are budding off of the ER. And what they're going to end up doing is fusing to either other vesicles or to the cis side of the Golgi apparatus. So here we can see how motor proteins are going to keep part of the Golgi's kind of anchored or moving along there. So, if we don't have movement of those motor proteins, we're going to be able to keep that Golgi present in one spot. And you can see how we have our COP2, our coat protein 2 vesicles are going to be moving towards the Golgi because these vesicles contain material that has been correctly folded. And so it's going to bud off and then move and fuse in with the cis side of the Golgi network. Okay. At the same time, we're going to be having parts of the Golgi budding off and being retrieved and recycled and go back to the ER. Okay. So the ER is kind of maintaining its size because we have vesicles budding off, right? So they're taking membrane with it. But at the same time, we have a retrieval piece to that is bringing material back to it. And so depending upon the function of the cell, that's going to dictate how large the ER is, right? So we saw in an earlier table how we had the hepatocytes versus the pancreatic cells. And so pancreatic cells are going to have a larger ER. So they're going to have more material because they have to make lots of protein. But they're always going to kind of keep that. There's always like an equilibrium of these vesicles leaving and coming back. And you can see how the coat protein has changed, and that's how it knows what direction to go. So this is just another figure showing the same thing. I kind of like this figure a little better because it's got some proteins in there, right? So we can see these yellow proteins that are going to end up being secreted, where we have these resident proteins. And so that if they get accidentally transported forward in the forward pathway going from the ER to the Golgi, they would be retrieved back and returned to the ER because we would have KDL receptors present in the Golgi, so that we're not, we're making sure that any of those residents that did escape come back into the ER. Okay, and again, another important piece on this figure is that cis side being on the ER side, where the trans side of the Golgi is going to be on the plasma membrane side, the piece that would end up vesicles that could undergo exocytosis. And in between there, you have the Golgi stacks. So we were always told in undergrad, it looks like a stack of pancakes. And so you can see in this figure on the bottom, we have our stacks of pancakes present. I don't know. My pancakes don't look like that. I want nice fluffy pancakes, not flat pancakes. A little bit more like crepes maybe. But you can see how, again, the cis side is on the ER side, where proteins are going to be newly synthesized, folded, and then they're going to be brought in. So just be aware that there are different theories on how material is moving through the Golgi. And so the kind of more accepted theory is that we have vesicles that are going to be moving from the ER to the Golgi from the cis side, but then from the cis, it's going to then move through the stacks and then come out through trans. The kind of other theory is that it's all vesicles moving, budding off between these different stacks, and so that the material that is going to be moving from cis medial to trans would be doing that through vesicles along the edges. And again, the more accepted theory is that you're going to have movement from the cis, and then it's going to move. The stacks are always moving. So it's not that the Golgi is static and staying the same, and it's just these vesicles that are changing on it. It's that what was cis becomes medial, what's medial becomes trans secretion, and then bringing it back through the retrieval pathway. Okay. So as proteins move through from the cis side to the medial to the trans, there are changes that happen to those oligosaccharides, the glycosylation that has happened to it. So there's additional modifications that happen, and I'm not expecting you to memorize like, okay, well, what are the modifications that are present on the cis Golgi versus the trans Golgi? Just be aware that there are differences and that this is very sequenced, right? And so we could look at a protein and look at the modifications on it and let us know where it is in that processing. So we wouldn't necessarily have to have like an intact Golgi to see where it was. If we just take protein and isolate protein and look at the modifications, that would let us know where this protein's journey is. Okay. So next we're going to look at Golgi to lysosome trafficking. And so lysosomes we categorize lysosomes as a number of different types of vesicles. So we can have late endosomes, endolysosomes, and lysosomes, and they fall into the lysosome category. There's slight differences between them and their quote-unquote maturity. But lysosomes, their characteristic is that they are able to break down nutrients. If a microbe, like a bacteria, has been phagocytized, so if it's been undergone the process of endocytosis, and if it's inside the cell, or if we have cellular debris that's been put into a vesicle, we can get fusion of these vesicles with the lysosome that allows for the digesting of that material. Okay, so lysosomes can fuse with late endosomes, and again, endosomes can be where material, cellular debris is going to be put. For if it's in a vesicle that's formed because of phagocytosis, it would be called a phagosome. And again, a phagosome would fuse with the lysosome and allow for the digesting. Okay, so there's not distinct markers. It's a little bit of looking at the structure to try to determine what it is and whether the material's been digested or not. Another way to kind of tell is there's mannose 6-phosphate that is on lysosomal enzymes. So if you were able to detect mannose 6-phosphate and you found that on the enzymes, that would indicate that you have a lysosome. Okay. In the figure to the right, you can see the different enzymes. So there's nucleases to break down DNA, nucleic acids, proteases to break down proteins, glycosidases and lipidases, phosphatases. So we have all of these different enzymes that are going to help to break down the material. These enzymes work best in an acidic environment. And so one of the features that are present in these vesicles is that they're going to have a hydrogen pump to pump hydrogen in to create this acidic environment. So the cytosol is going to have a more neutral pH, right? But these enzymes break down material more easily at an acidic environment. Okay. And so again, we can see here how with endocytosis, we have material being brought in. So the endosome is present there, but if you go back to like another figure, we can see that material can enter into an endosome from like the Golgi or from the ER. So endosomes would be produced. Maybe we have like damaged organelles where we have autophagy. So here we see an autophagosome that has material from the cell, cellular debris. We also have a phagosome. This is where we have like a bacteria that's undergone, that the cell has undergone phagocytosis to and engulfed it. And again, that vesicle would be called a phagosome. And so these will fuse with the lysosome with these digestive enzymes, and that dissolves this material. Okay, so again, lysosome will fuse with the vesicle. The material in the vesicle will be digested because of the enzymes present in the lysosome because of the acidic environment. And that means that that material has been destroyed. The enzymes, the material can be neutralized and then released and recycled for the cell to use, or if it ends up going through exocytosis for other cells to use. So in microbiology, if you take the course, you'll learn that some bacteria actually have ways to stop the fusion of the phagosome with the lysosome, and so they can actually replicate within the phagosome. So again, I think I mentioned rickettsia. Rickettsia would be an example of one that has that ability to do that. Okay. So for some receptors that have been introduced into like a vesicle through the process of endocytosis, they can be recycled. So what we're seeing here is we've got endocytosis. We have this vesicle forming. It's going to develop, it's going to end up fusing with an endosome. It hasn't fused with the lysosome yet. So this is the point in which if receptors are going to be recycled, that they need to be returned to the plasma membrane. So this is where we can get budding off of vesicles so that they can undergo like basically exocytosis and return to the surface. If they are not returned and you get the fusion with the lysosome, you're going to end up destroying the receptors. So again, it's important that anything that needs to be recycled before the fusion with the lysosome happens, that material is moved on. So we've been talking about vesicle formation, and so for endocytosis, this is where we are going to get transport into the cell from the plasma membrane, right? So we're moving from the cell surface, the plasma membrane, and then we're bringing material into the cell, and we could bring this in as an early endosome, or we could be calling this a phagosome, and that material could end up going to the Golgi, or it could be developing into a late endosome, where then it would be fusing with the lysosome. Okay, the process of endocytosis, there's specific terms depending upon what cell is doing it and what material is being taken up. So again, endocytosis is this general term. Phagocytosis is when large particles are being taken up, and this is usually a term that's used with immune cells. So white blood cells, leukocytes, they can undergo phagocytosis. So your neutrophils could undergo phagocytosis, and this is Latin for cell eating. So it's when a cell is going to eat another cell. This process is also done not just for foreign material, but also for damaged cells. So in the image below, we have this phagocyte, a cell that undergoes phagocytosis, engulfing red blood cells. And so where the arrows are indicating, that's actually where the pseudopodium, the processes from the immune cell, are coming up, and they're going to end up engulfing that whole entire red blood cell. The red blood cell would then end up fusing with a lysosome, so it would be digested. Okay, but again, phagocytosis is where like a whole cell, large material, is going to be taken in. Pinocytosis is where we get this ingestion of small amounts. So usually liquids, and then there would be like proteins and other molecules that would be present in it. And this is known as drinking. So the cell is drinking the fluids around. Okay. So there's also turgocytosis, which most textbooks don't talk about. Turgocytosis is where membrane is taken. So it's not phagocytosis because the whole cell's not taken up, but some of the membrane and any of those surface receptors are taken with it when a cell undergoes turgocytosis with another cell. So it's actually a way in which cells can sample other cells. This process has been studied in cancer. So immune cells will undergo turgocytosis with cancer cells, which actually teaches the immune cell not to respond to the cancer cell. So it's a cancer cell trick they have. They let the immune cell take a little piece of them, and it's enough for the immune cell to say, that's kind of me, right? Because your cancer cells are you, and they say, oh, we shouldn't respond, move on. But the cancer cell is not actually damaged because the membrane's fluid, right? And it just kind of heals itself. So I only know about turgocytosis because I looked at turgocytosis as a process in prostate cancer cells, and that immune cells can undergo this process. And for aggressive forms of prostate cancer, GRP78 actually acts as a target to tell the immune cell, try this part of the membrane, because it actually brings damaged unfolded proteins to the surface in aggressive forms of prostate cancer. And so in this process of doing that, the immune cell takes parts of the prostate cancer cell with it, the membrane, the GRP78, the damaged proteins, but it actually doesn't end up responding. And so the trick then becomes, how can we actually make the immune cells know they're being tricked and respond anyways? So anyways, fun facts. little added information. Turgorocytosis probably won't be on the exam because it's not in most textbooks. So the other piece is exocytosis, right? So endocytosis and exocytosis work kind of together. Endocytosis is bringing material in, exocytosis is releasing material. And so this is, we're going to have material transported from the Golgi out to the cell surface so it can be secreted. And that process can either be a more direct pathway, or we can have vesicles involved, secretory vesicles. So cells have two different pathways, secretory pathways. They have the constitutive secretory pathway and the regulated secretory pathway. With the constitutive pathway, this is unregulated, and this is where just membranes are going to blub off with synthesized proteins, and then they're going to fuse with the membrane. In the regulated pathway, there is going to be signaling that feeds back to tell the vesicle to then fuse. Okay, so this is where we would have cell signaling, hormone or neurotransmitter would bind, and then that initiates the secretory vesicle to end up fusing. So, again, thinking about our neuromuscular junction where we have the neurotransmitter coming across, binding, signaling for calcium to be released so that we can get muscles contraction. Where there's going to be certain molecules that will end up getting secreted because, again, they have an exit signal, but there's not the regulation to it, so they're going to be constantly being released in that process. This process of exocytosis is going to be regulated by SNARE proteins. So same thing, but kind of in reverse, right? So that we saw with the endocytosis. So we're going to get that docking. So we're going to have our tethering occurring, and then we're going to get the snares forming so that we can have fusion of the membrane. Now, instead of this fusion, this connection allowing for material to move from like the lumen of the vesicle to the lumen of the Golgi, now we're seeing where that fusion, that connection allows for the secretion of the material that's inside the vesicle to outside, right? Because the membranes that are making up the vesicle are going to become the plasma membrane, okay? And so we can see here below how on the left-hand side on this electron micrograph, we still have the vesicle membrane and the plasma membrane. They're not fused yet, but then when we look on the right, the material, the membranes have fused, and now the material that's inside of this vesicle is free to be released. So an example of how of exocytosis in cells, I had mentioned with neurons, the neuromuscular junction, how we have the calcium release, right? But also we could have like the mast cells. Mast cells are an immune cell that releases histamine. And so we can see here we have our mast cell before histamine's released, and so we can see all these vesicles that contain histamine. And then once the mast cell is signaled to release, all these vesicles end up fusing with the plasma membrane, and there's this massive release of histamine, right? So if you have like allergies, this is the process that's happening to your mast cells, where we end up getting this histamine release. Exocytosis can also allow for the cell to become enlarged, so it increases the amount of plasma membrane that's present, right? So if we think about it, there's a lot of processes that are going to cause whenever we have endocytosis, we're going to end up having a decrease in plasma membrane. So we need to make sure that we are also having vesicles fusing with the plasma membrane to restore the amount, right? So, again, examples when we have cell division, and so we have that cleavage furrow, when we have phagocytosis, when we have a wound, those are all going to cause there to be a decrease in the amount of plasma membrane available. So we need to make sure that we increase the amount, like go back, like we have like 100% of the plasma membrane when we undergo phagocytosis, or we have a wound, that's going to decrease. So we need to make sure we have vesicles fusing with the plasma membrane so we can increase that the amount of membrane that is present. Okay, so this concludes the slides for chapter 15. There's a lot of information in there. I cut out a lot of the slides just because I wanted to make sure there wasn't this over information. If you have any questions about the slides that I removed, please do ask. I'm more than happy to use our collaborate time or class time in order to go over those. But there's a lot of content without them. And so given the fast-paced nature of the course and all the other material, I do think some of those slides might help understand, but it might also add so much detail that it also causes confusion. So I would strongly recommend, definitely, if you have any any questions, understanding any of the audio lecture slides and information for chapter 15, definitely reach out, send me an email. Make sure that I go over it in class this next week. But again, if it's on the slides I've taken out, I've chosen to take them out for a reason. Okay, so thank you very much, and I hope you enjoyed the audio lecture.